right. Welcome back to the 2025 Borealis Psychedelic Science Summit in Stockholm. My name is Yoshi, and I'm joined now by Professor Gitte Moos Knudsen. Welcome.
Thank you.
Thank you so much for sitting down to chat with me. We are in day 2 of the conference. How did you enjoy day 1?
I thought it was a marvelous day. Very nice. Very good atmosphere and relaxed and good speakers and so on. Yeah.
Did you catch a lot of the lectures, panels?
I did, yes.
Fantastic. Tell me a little bit about your background, about how you got to working in the psychedelic field.
Yes, it's an interesting story, as it almost always is.
Yes.
But I have been my background is a neurologist. And I trained as a clinical neurologist, but for the last couple of decades, I've been running the Neurobiology Research Unit at the University Hospital, Rigshospitalet in Copenhagen, Denmark, where we do many types of different translational studies. So it covers from animals, cell studies even, to healthy individuals and also involving patients with either neurological or psychiatric disorders. So it's really quite broad interest area that the research unit is focusing on. Mm-hmm. And in particular, what we do is neuroimaging of modulatory effects on the brain. I'd like to put it like that. Mm-hmm. Back in 2005, I was fortunate enough to receive a major grant from the Lundbeck Foundation, not to be mixed up with the company. That allowed us to establish the Center for Integrated Molecular Brain Imaging, where we started to work on the serotonin system. And one of the things at that time that was very much discussed is whether we would be able, by means of positron emission tomography, to measure endogenous brain release of transmitters. Mm-hmm. And at that time, we had a tool called raclopride that enabled you to measure in healthy and patient studies in alive people the release of dopamine in the brain. And the big challenge was then, could we develop a tracer so we could also measure other neurotransmitters? And I was particularly interested in serotonin at that time, and still am actually. So we embarked on developing a serotonin 2A receptor agonist radioligand, because at that time it had been shown that agonist radiotracers were more amenable to competition with endogenous neurotransmitter. And we succeeded in doing so, and in 2014 we published the paper on a radiotracer called SYMBI-36, mm-hmm. which is this serotonin 2A agonist radiotracer. And then, of course, we started to use it for various purposes, and we could indeed determine that it was sensitive to endogenous release of serotonin, except that you had to have a major elicitor of serotonin to see that difference. And then at some point, we started looking at the classical psychedelics that all activate the serotonin 2A receptor. If you block the serotonin 2A receptor, you have no psychedelic experience. Mm. And that would include compounds such as psilocybin, LSD, and DMT. So I set up in a later study the occupancy studies, where we can look at various doses of psilocybin and how they interact with brain serotonin 2A receptors. And in that way, we were able to, in 2019, show how increasing doses of psilocybin were associated with, of course, increasing doses in plasma psilocin, which is the active metabolite. And not only that, you also could see an increase in the subjective experience, but that they were fairly nicely correlated. And we could also show that plasma concentrations correlated in the expected manner with brain occupancy at the 5-HT2A receptors. And that was kind of the beginning of doing psychedelic studies. And So we have moved on from there. But that's the story about how we got into this area.
Why would someone want to block the 2A receptor? Is it to get the benefits of, for instance, psilocybin without getting the psychedelic effect, or what is the purpose of doing that kind of research?
Well, I mean, the reason that people have looked at the effects would be to say something about the specificity of the nature of the psychedelic experience. So the interesting part is if you are able to block the psychedelic experience by giving a 5-HT2A blocker, then we know that this is the receptor which is responsible for these features and the psychedelic experience. And very little is left. So, of course, most of the well-known classical psychedelics do not only target the serotonin 2A receptor, they also target other receptors, other serotonin receptors as well. But it does not seem to have a major impact. It's really the 2A receptor that is driving these psychedelic experiences.
Right. What's something that you're most excited about in your current work?
So now we are becoming more interested in the mode of mechanisms. Is it because we have a subsequent downregulation of the serotonin 2A receptors? Doesn't seem like that. We looked at that. Some people talk about TrkB being involved and the brain-derived neurotrophic factor, and that is generating neuroplasticity. So we are on the hunt for a good PET radiotracer for the TrkB and also for measuring BDNF in the brain. But we have also completed a study where we look at a particular neuroplasticity marker of synaptic density. We've seen in the pig brain that psilocybin is associated with an increased neuroplasticity in terms of synaptogenesis 7 days after the intervention, but not in the acute phase. So we have done this study in humans where we looked at the synaptic density after 7 days after a single dose of psilocybin at a psychedelic dose. And we can see that the change in synaptic density seems to be contingent on the setting in which the person was in while they got their psychedelic experience. So if they're in a noisy MR scanner, seems like they have less of neuroplasticity compared to being in a more gentle environment with soft music, etc.
Interesting. Are those two things linked? So does neuroplasticity lead to higher synaptic density?
Well, I mean, that's what we can measure at least. So,
right.
So of course you can always argue what is functional neuroplasticity, what is structural neuroplasticity. But in this case, we are looking at structural neuroplasticity in terms of number of synapses. Wow. But you can also measure in other ways, of course.
Right. And you said for 7 days after the experience,
yes,
the density is still higher.
Yeah.
That is so interesting.
Yeah.
Yeah, so it actually leads to a more developed brain in some sense.
Well, I mean, I don't think of it as a more developed brain. I think every time you use a certain pathway where one neuron is signaling to another neuron, you strengthen the connection between these two neurons.
Right.
I think we would rather like to think of it as psychedelics changing your brain in a more positive manner. So my guess is, but we are looking more into that, hopefully soon, is that you can see an increase in the synaptic density in the beginning, but then that will probably be followed by some pruning of less efficient synaptic contacts. So I think the most likely pattern would be to see an increase in synaptic density that is then followed by a gradual reversal to maybe what it was but now it's some other synapses that have been strengthened, so that networks that have been not very positive are then being pruned out and new, more healthy connections are being made.
Interesting. And you mentioned that in a more gentle environment, soft music, this effect is stronger then.
Yeah, we have an interaction effect. So we see that if you are in the gentle environment, this seems to be more. We'd like to do a larger study to confirm this, of course, but I think this is an interesting finding, how setting can interfere with what is happening.
And this is with experiments on pigs, you mentioned.
This is what
experiments with pigs.
No, this is in humans.
Oh, this was in humans.
This is in humans.
You did mention
we both did in pigs and we did in humans.
Okay.
But the last one is this.
Okay.
Yeah. No, the pigs were not in. They were in the same environment.
Okay, got it. That would be interesting to see if other animals also appreciate the soft environment.
Yes, I mean the whole debate about what animals do and how do we know if they have a psychedelic experience is of course quite interesting. So rodents have a particular behavior called head twitch response, where they shake their heads, and that is thought to be representative of a psychedelic experience. Pigs do also shake their heads. Do they have a psychedelic experience? I don't know. Yeah. But the nice thing about the occupancy study with PET is that now we know what dose to give the pigs to have comparable occupancies in the brain compared to humans. So we know for humans, if you have 60% of occupancy of psilocybin on the 5-HT2A receptor, you have a profound psychedelic experience. So we know how much to administer to the animals because we've also done PET studies in the pigs.
Got it. Is this what your presentation during this conference is about?
During the conference, I covered more broadly some neuroimaging data that we have, and some of them I've already talked about here. But on top of that, we have also done LSD occupancy study that we recently completed in humans, in healthy humans. Where we gave LSD, different doses, put them in the combined PET-MR scanner and scanned them to determine the occupancy levels. And we took blood samples to determine LSD concentrations. And we also looked at cerebral blood flow and functional connectivity. And what we found is, not surprisingly, because we know that already, that LSD takes more time to set in when you take LSD. It takes a longer time to elicit this psychedelic experience. And we knew that kind of already.
Compared to psilocybin, you mean?
Compared to psilocybin, where you have a 1-to-1. So if you have a plasma concentration, you also have a subjective intensity that is responding. What we saw here was that as plasma LSD starts to increase, we have very little psychedelic experience. But then at some point, when it starts to return and get lower again, then the psychedelic experience sets in and increases. So there is kind of a delay between what's happening in the plasma and the blood and what's happening behaviorally and psychedelically.
Interesting.
Yeah. So that was not so surprising. We could also see that we also needed, with this compound, about a 60% occupancy in order to have a profound psychedelic experience. Interestingly, we have seen for psilocybin, we and also others before us had shown that the cerebral blood flow goes down with psilocybin administration. What we saw with LSD was actually the opposite. And for a while, we've been wondering about the impact of these compounds on blood vessels to the brain. And we now know that when you give psilocybin, you have a constriction of the arteries going to the brain, which potentially is associated with a reduction in the blood flow to the brain. Now, with LSD, we don't see this constriction, probably because it takes longer to set in, so it's a more slow and gradual effect. I don't know. and we see an increase in cerebral blood flow. So we have opposite effects on cerebral blood flow, which is a little bit puzzling. And we also see opposite effects on functional connectivity. That is, what parts of the brain are talking together. And traditionally here we've seen with psilocybin that the brain starts to become much more interactive. So rather than just being stuck in the usual 7 canonical circuits that are running, psilocybin makes this change so that the brain, so to speak, becomes much more interactive. And now, I'm just wondering, is this a methodological thing we are seeing with these MR scans? But that is an interesting question to pursue in the future.
A lot of work left to be done.
Sure enough.
I think that'll probably be the case forever and ever in this field. Speaking of the field, tell me about something in general in the broader psychedelic research field that you're excited about today or moving forward.
Well, I think what we're seeing now are the large clinical trials, so phase 3 trials. The field has generally suffered from data. I mean, scientific data. We have a lot of data from people who have taken the drugs. And of course, it makes you always wonder, how certain are you that you're actually taking the drug that you think you are getting? We know that it's not always the case. The doses associated, we don't know that. So I think there's been a lot of storytelling too. But now with some of these large clinical trials, we'll get much more data and much richer data and much more well-controlled data. So I think that is something that will be very helpful for us to understand a lot of things much better.
For sure. Where can people find and follow your work?
Well, we publish, of course, in scientific journals, but if you go to www.nru.dk, you can find our website.
Fantastic. Professor Gitte Moos Knudsen, thank you so much for sitting down and having a chat with me.
Thank you for the invitation to come.
Much appreciated. And thanks for everyone tuning in. My name is Yoshi from the Borealis Psychedelic Science Summit. Catch you next time.